very grateful to the rector to host us here at this wonderful location, to Jenner and his colleagues who organized perfectly this symposium. It's really impressive. My pleasure to be here. And I have to admit, it's my first time ever in my life in Turkey and or Istanbul. So long overdue, hopefully the first of many visits. And that gives me the opportunity to clarify something I've been struggling with for all my life, because our family name, I've been told, could have different origin. It could come from Czech Republic. It could also come from Turkey. And I was told the translation of Tschöp in Turkey is less flattering because it means trash. Is that correct?
Yeah.
Well then, that is settled. Nevertheless, I'll try to present something that's not trash today, but I put together the history of these obesity drugs, some latest developments, and I also put in an unpublished paper that's currently in review, so hopefully there's something for everybody. I mean, even for those of you who are working in nephrology or other things, as I heard, you may have noticed there's something going on with obesity. There's all these reports, and this is the one that Jeff earlier referred to today, the everything drugs, whatever that will mean. But there's a lot of excitement that the new obesity drugs, these weight loss drugs, are changing the world we live in or the way we do medicine, at least to some extent. Now it really comes down to a family of drugs, of therapeutics that is summarized here that are gut hormone based, mono, dual and triple agonists. And Jeff and I sat down for a couple of months and went through the history of the discovery of these obesity drugs. And as you can tell, nobody can or should read this table now, but this has been a long process, step by step, incremental progress. This didn't happen overnight. Now this journey started for these drugs I'm going to tell you about today, and for me personally, in this building at the University Hospital of Munich, 31 years ago. And I saw a lot of patients with obesity, diabetes, and thought, well, this is a really important problem, and we could prevent type 2 diabetes largely if we would find something that helps us to normalize body weight, but we can't. Now the numbers were going up and are still going up for obesity worldwide. Obesity certainly comes with a bunch of comorbidities that are important to look into. It increases risk for cancer, cardiovascular disease, etc., etc. Or as a very simple message, obesity kills. These are already outdated numbers from the WHO saying a couple of million people die every year of obesity. So if we had a drug that would allow us to regulate body weight significantly and safely, we could save a lot of lives. Now that was of course attempted for hundreds of years with sort of not a lot of success. At the time when I was as a young doctor seeing these patients, this was the situation. There were therapeutics that were not safe, others that were not efficacious. It was never really going beyond placebo effects, or one would run into problems of toxicity. So I thought, well, this is just the right thing for a young endocrinologist. I'm going to devote my career to finding these types of drugs. Now, that was enthusiastic, also quite naive, because there was no molecular basis of body weight regulation known at the time. Luckily, then in the year later, in 1994, something happened and Jeff discovered leptin. So now a bunch of things happened. We first of all knew there is a molecular basis to body weight control. This is actually possible. There might be other signals. And while it was figured out relatively quickly that just making and giving leptin wasn't the only solution, or not the quick solution for obesity, this really set the field in motion and gave us lots of opportunities to pursue these novel drugs. Now, I remember very well the moment where I read the paper in our hallway in our hospital when that came out. And coincidentally, there's photographic evidence of me sitting there looking at that paper. Now, look a little bit pre-aged for 27, but notwithstanding, that is the actual paper we looked at. My friend Martin and I went through this and said, well, exciting. It's a new endocrine organ, the adipose tissue, there's a new molecule, leptin, and there's a whole new mechanism. Apparently this works in the brain. And this, I think, is one of the most important things we learned, that this important regulator of body weight acts in the brain. And that drove partially also my intention then to find out more about this when I went to Eli Lilly, into the research labs in Indianapolis, and looked into maybe other players, and we found that there is a peptide from the stomach called ghrelin that is sort of a leptin opponent. It's a hunger hormone, it increases appetite, etc. Does the opposite of what leptin does at most of the same targets in the brain. Again, a disappointment. You couldn't just block ghrelin and do a whole lot, but it became more and more clear that, well, the brain is important, and there are several signals that are regulating food intake and body weight. Now, a lot of that was mouse research, and luckily, Steve, who's here today, Steve O'Reilly and colleagues, and Sadaf and Giles, who's not here today, figured out a lot of things that had to do with translating this into humans. So first of all, leptin also is relevant for body weight regulation in humans, and if you don't have leptin and you're a human being, you're in trouble. Body weight will go up and you will get diabetic and so on. And if you replace it, you can rescue that phenotype, which was one of their big findings. But they also made contributions, as Sadaf and Steve will tell you later, that point to the fact that the brain is really important for obesity. You might as well, if you simplify things, go as far and say obesity is more of a brain disease than an adipocyte disease. And many others, the whole field figured out, and I think it's important to say for obvious reasons, that obesity is mostly a genetic disease. It's not a character flaw. Of course, it has to do with environment, gene interaction, all of these things, but it is something that is a serious disease like other genetic diseases that we know about. So if you're a young physician like myself back then, you want to make drugs, you need somebody who actually makes molecules. So I was lucky to meet, while I was at Eli Lilly, Richard DiMarchi, fantastic peptide chemist, had already made a couple of drugs. And we together said, okay, we're both going back to university, Indiana University, Cincinnati University, where I worked back then, and we'll figure this out together. We'll try back and forth, a chemistry lab, a physiology lab, pharmacology. We're going to be making drugs in academia. And we had a clear strategy. You need to target the brain. You don't want to do this directly, as we have learned from cannabinoid receptor antagonists and others. If you mess with signals and neurotransmitters directly in the brain, you'll frequently have side effects. And pharmacology just isn't as far yet that we can directly target only a few neurons or synapses or places where we want to go. So we were going to use a peripheral system, sort of Mother Nature's toolkit of gut hormones, and these hormones knowing very well which neurons they need to target, modulate those, and do it in combination. Go beyond one signal, 2, 3, 4, maybe more signals. And there was an exploding pool of potential signals to look into. We actually tried a lot of them in sort of trial and error approaches. We also took a little bit of a look at what surgeons were reporting, how right after the surgery of putting a gastric bypass in place, some of these signals are changing in certain directions. So we thought, okay, there's maybe an indication which ones matter to successful body weight control. And we ended up really looking at the family of glucagon hormones. Now, glucagon, most people that I knew back then said, is the wrong hormone to look at if you want to activate a receptor, because you drive up blood glucose, hyperglycemia. You don't want that. But we said, well, there's a bunch of other stuff that glucagon does. It drives energy expenditure, it drives lipolysis, it helps with decreasing food intake. So glucagon is a perfect anti-obesity hormone if you forget for a moment about the diabetogenic liability that it brings, which you may be able to override with something else, because we were thinking about combinations of molecules. Now, when you're a peptide chemist and you look at this family of hormones, glucagon-like peptides, GIP, you notice that there are some similarities. And Richard thought he could engineer one of these peptide hormones, change a few amino acids, do a few things differently, and turn it into a superhormone. It can activate 2 or 3 receptors. Now, you don't want to change too much, because you then cause your own body to not recognize this molecule as something that's likely natural, and you have immunogenicity, and you destroy not only the drug, but maybe your own hormones. So that was what we were going to be working on, and we started out by actually making agents that you can use for these kind of experiments to test and figure out what works in cells and in mice and in rats and later in humans. And for that, these gut hormones are not very well suited because, as Jeff had mentioned this morning, they have a half-life of what, 2, 3, 4, 5 minutes, depending on which one you're looking at. Now you want to have versions that are longer acting. And that's one of those stories that were reflecting incremental progress. A little change here, a little change there were made, and after a while, there were more and more molecules available. Now, that was mostly true for GLP-1. For glucagon, nobody really cared because nobody really wanted to enhance glucagon. So we were the only ones that were working on this and said, well, we now know from GLP-1 research, I think this is not quite working, that there is this guy here, Heloderma horridum, and the name probably comes from the fact that if it bites you, it's horribly painful because it has a toxin, a venom in its saliva. But it also in its saliva this lizard has a version of GLP-1 called exendin that is more stable. And it has that CEX-talase, as we call it, that is this part of the peptide that helps it to be longer acting, as a longer half-life, still soluble. And Richard thought, well, we can put this on glucagon and make that longer acting and more soluble, which we did and tested it. It infused it into mice that were obese because they have been for 6 months on sort of a McDonald's diet. And we could decrease body weight significantly. This was one of those moments where you're like, ooh, this is actually real, right? 2006, we see you can use glucagon to decrease body weight. You can also use exendin, GLP-1 receptor agonist, to do that if you give enough of it. Back then, these molecules were not as refined as they are today. But it's two different systems, both being very successful, so we thought this is the basis for our coagonist, for our superhormone. Let's try to make a glucagon GLP dual coagonist. Now we started talking about this, as you do in scientific communities, to your colleagues and peers and industry and reviewers, and everybody said it was very clear that we were full of it, because you can't enhance glucagon receptor action. This is going to backfire. You're never going to get funded. You're never going to have a drug. You should forget this now. Of course, we were a little stubborn, and we had some good data that we believed in, and we concluded, and still conclude today, now there's a lot more people that believe in this, that glucagon receptor agonism, not antagonism, is helpful for fighting metabolic disease. Now at the time, there were lots of pharmaceutical companies that were actually blocking the glucagon receptor. They were looking for antagonists, and they had antagonists. Turns out they had side effects, lipid metabolism, etc. So these programs were stopped while ours then continued. So now we had glucagon, we wanted to make dual agonists, and started by making molecules in Richard's lab that had both capabilities, activating the GLP, activating the glucagon receptor. And it's important that you have the right balance, right? You don't want to have too much glucagon because of the blood glucose, and you don't want to have too much GLP only because you need to enhance it with something else, I thought. Now talking for a moment about GLP-1, another gut hormone that I clearly did not discover or make massive contributions to, but this was the work of Joel Habener, Svetlana Moisov, and others, who probably were the first to discover the bioactive sequence of GLP-1 in 1986, 1987. And they have a patent on this in the first publication and just received the Lasker Award. And I may mention, just received another award, the Princess of Asturia Award, which will be on a picture that comes up eventually. The next slide is sort of thing. Yeah, here we go. And you will recognize the third person here. Jeff also received this award last week for obesity research, obesity drug development, etc. Now, GLP-1 as a bioactive substance was discovered, as I just said, in 1987, but that was just regarding its impact on insulin secretion. That's why it's called an incretin. Now, nobody then cared or knew about its potential effect on food intake or body weight. That came much later through work by Steve Bloom and others that put actually GLP-1 in the brain of mice and rats and figured that there is a decrease in food intake. Here we go. So that was the first paper showing that by Steve Bloom and others. And then Suad Effendi, actually, together with Richard DeMarchi, were the first ones to show in humans that GLP-1 can decrease body weight, and that became a patent that, of course, now has long expired. Now, GLP-1 seemed to be an obvious candidate. At the time, we knew it was doing something to food intake and body weight in the right direction, but it was sort of a limited phenomenon still. And we thought if we enhance glucagon and GLP-1 mutually, then we have something, and that turned out to be right. So these are preclinical data in mouse models of a first dual agonist for gut hormones, glucagon and GLP. And depending on how much glucagon activity you put into the molecule, of course we tested a number of those, you can drive down body weight by 20, by 30% in these obese mice. Now, the big question was, are we doing something bad to glucose metabolism? Not really, as it turns out. We're improving glucose tolerance, and we're improving insulin levels. So that seemed to go in the right direction, because you have GLP, you decrease body weight, and long-term glucagon action may not be the same as short-term glucagon action. So this got published. I have problems getting to the next slide in time. I don't know. It's hanging. This got published in a journal 15 years ago, and today we know this works clinically as well. There's a number of dual glucagon GLP-1 receptor agonists in phase 3 trials. This is one version, survodutide, of Boehringer Ingelheim in Germany, but there's other companies that have similar versions. And it allows already in phase 2 to drive down body weight 18-20%, may go a little bit higher when we get the phase 3 data. So this works in humans. This was the first one. Now, of course, we thought we've seen a lot going wrong in obesity research and drug development. We need to have different versions, right? So we need to have different shots at the goal. So let's look at another incretin, GIP. What if we have a dual agonist for GLP and GIP, two incretins that both definitely have benefits for glucose metabolism, may also have benefits for weight control. Now again, you make a bunch of molecules first to understand GIP better. Short half-life, not a lot known about its physiology. So we made a bunch of these agents, tested them in cells, in mice, and rats, and first of all made sure, yeah, it does what it's supposed to do, it decreases blood glucose. It's an incretin, fine. So we did something that seems like it might be a GIP. Now the next question is, does it do something to body weight? Does it make sense to continue? Yes. These longer acting GIP receptor agonists decrease body weight. And the third question was, well, or maybe we did something else here. Is this really specific? Is it a GIP agonist or is it a GLP agonist? And for that, you can really use knockout mice very efficiently. And we could show that, yeah, in the GLP-1 receptor knockout mouse, the GIP agonist works well. And if you knock out the GIP receptor, it doesn't. So we're clean and specific. And these are example experiments, what we did dozens of times, if not more. Again, when we started talking about you need to activate the GIP receptor, this is what we got. Everybody said, you're totally wrong. You need to turn around. You need to block the GIP receptor. Everybody knows that. And we thought that was not the case. Our data were speaking a clear language. So we continued working on this. And today we have seen clinical data, for example, out of Eli Lilly, with a GIP receptor agonist, specific activator, it decreases body weight. So it doesn't increase body weight as was feared at the time. Now, this is the second take-home message. It's GIP agonism, like glucagon agonism, that overall may be the right thing to put into these dual and triple agonists that you're hearing about. Now, making a dual agonist of GIP and GLP again took some time. You need to figure out the balance. We have different versions. They need to work in mice, then they need to work in rats, so that in the end you have something you can translate. But this is our first version, dose-dependent decrease in body weight possible. We published it about now 12 years ago. And we actually did the first clinical study with a GIP, GLP dual agonist. This was the first of its kind. And a post-hoc analysis shows even after 12, 13 weeks, this was better than monoagonism, so as a proof of principle. Now, today, everybody knows, most of you will know that there are other versions of GIP, GLP dual agonists. One of them is Mounjaro or Zepbound or tirzepatide by Eli Lilly and Co. And it's called the biggest drug ever, if that's true or not, but it certainly makes a lot of money for Eli Lilly, and it helps a lot of patients. And the transformative breakthrough here really is perceived as this is the first drug ever that allows to decrease body weight on average by 22-23%. It can normalize body weight in significant cases of obesity. And those of you who are not working in that field, this is what that means. This is what this looks like, and this is what this means to these patients. You really make a difference. Now, let's not forget that these drugs, they're incretins, they're triggering insulin secretion, they're improving metabolism, also help a lot with fighting diabetes. Over here, these bars, they indicate that in the higher dosing of Mounjaro, you can achieve more than half of the patients with type 2 diabetes that are treated having normalized blood sugar, which is an enormous success for something that is not insulin. I believe important are views of the side effects, because GLP-1 based drugs like monoagonists, Ozempic, Wegovy, they come with side effects like nausea, emesis, gastric emptying delay. And you have these with dual agonists in a comparable manner, but you have significantly improved benefits. So relatively speaking, you have a wider therapeutic window, fewer side effects, less side effects, and comparable action. Now you can test these things, these side effects, in a preclinical model. Normal mice and rats don't do that well, but this musk shrew actually can vomit, which is helpful in this case, because then you can study these drugs and you see, well, this is what GLP-1 does. It increases vomiting. GIP doesn't do that. If you combine these two, you actually suppress the side effects of GLP. And I personally believe that that is one of the advantages of dual agonism, that you suppress some of the GLP-1 side effects, and you actually have fewer side effects, and you increase the benefits by adding GIP. This is, I think, a less peer-reviewed data set, but I think it's really telling how the gastric emptying delay that's called frozen stomach that many clinicians report to see in treatment with monoagonists, you don't get that with dual agonists for the reasons I think I just mentioned. Now in our lab we're trying to figure out ways to better understand the mechanism of these drugs. We certainly believe this is happening in the brain. This is Timo Müller leading our lab in Munich, and he has clearly shown that if you remove the GIP receptors from the central nervous system, you lose most of the additional benefits that a dual agonist brings on top of GLP. Now we have been looking more closely into this and have been staining and identifying these neural populations that we believe are mostly located in the hindbrain. And when you remove the GIP receptors selectively from those neurons, those are, we believe, GABAergic neurons, you use a VGAT GIP receptor knockout model, then you lose the significant additional benefits. So we believe somewhere there is the real mechanism. Now, some unpublished data in that direction that are currently under review, and for those of you who have heard me talk before, it's the first time that I'm showing this. This is my grad student, Robert, again working with Timo and others. And we have been working with a question that is tricky in the field, because there's many companies now, many labs that are pursuing dual and triple agonists. There's one lab, one entity at Amgen, a pharmaceutical company, they say they have a combination of a GIP receptor antagonist and a GLP-1 receptor agonist, and that together works as well. It's a little bit different in many ways. They have an antibody-based entity, etc. They have a very high dose of GLP-1, and ratio is several hundred-folds higher than what others use with co-agonism. But there are results preclinically and in the phase 1 trial. Now, how can that be? You antagonize something here with GLP-1 agonism, you agonize something here, and both times you get the benefit you're looking for. Well, we think there are ways to explain this. So we have been trying to use some of our models and giving antagonists and antagonist-agonist combinations to this model And the same neurons that are responsible for the GIP receptor agonism benefits don't seem to be relevant here, right? We can't get rid of the effect of the antagonist when we test it in mice. Now, we actually looked at another model where we knocked out GIP receptors in the peripheral nervous system. We can't get rid of the benefits of antagonists against the GIP receptor either. So what is that? So let's go to global knockout mice. Here are the effects of an antagonist. If you knock out the GIP receptor globally, you get rid of the effects of the antagonist. You better, otherwise it's not a GIP receptor antagonist. Interestingly, when you only knock out the GLP-1 receptor and you give a GIP receptor antagonist, you also have no effects. So the GIP receptor antagonist requires the GLP-1 receptor to achieve weight loss. Now that's not the case with the GIP receptor agonist. We have tested that and we know that. So we have been digging around for the last 2 years, lots of single cell sequencing, hindbrain, hypothalamus, comparing expression patterns, etc. And there's a couple of interesting insights emerging. One is these antagonists against the GIP receptor, they are correlating and look way more like a GLP-1 agonist, while the GIP agonist looks more like MAR709, that is a dual agonist. So you can already tell that it's going a little bit in a different direction. This doesn't seem to be the same mechanism. So antagonists are like GLP, dual agonists more like GIP, and then there's all these fancy pictures where we see, well, there's hindbrain neuronal populations, GIP more widespread than GLP. Now I have to say, all of what you see here is mice, and I'm sure Steve and Sadaf will figure out how much of that is true in humans. So far, a lot of this has translated in the past. But final piece here, if you look at the gene programs implicated, then there are patterns, if you compare them, that again say, well, GIP receptor antagonism is engaging in cell populations and triggering gene programs that look a lot like GLP-1 receptor agonism, and the GIP receptor agonism adds something very specific to the dual agonism. So this, by the way, is hindbrain specific. We don't see these differences in the hypothalamus. It could be that the signaling from the hindbrain to the hypothalamus feeding circuits is what's modulated here differently by these two different approaches. So take-home message, we believe, is, and we sort of know through the rumor mill, there's another group who has the same or similar data, and that's also in review. GIP receptor agonism, antagonism engage different mechanisms to decrease body weight, and the additional weight loss induced by GIP receptor antagonism probably is based on a sensitization of the GLP-1 receptor, or enhancement of GLP-1 receptor signaling. Now, what I would be worried about if you just increase GLP-1 receptor signaling, that you also get more side effects, but we will see with more clinical data. All right, so much to that ongoing or recent study, and I close my pharmacology part by saying that, of course, when you have two dual agonists, you want to make a triple agonist. So for a while we thought, should we try this? Because it's incredibly complicated, right? You need to make sure you have the right balance of 3 entities, and you only have a few amino acids to work on. So it really becomes sort of a Rubik's Cube thing, but it's a lot more work than solving a Rubik's Cube. So it took us, I think, 5 or 6 years. Going through every amino acid of one of these hormones, of one of these compounds that more look like this, make sure they're protected against cleavage by enzymes, making sure they're linked to fatty acids or diacids these days, having that CEX tail maybe, or nowadays maybe even we don't use this even anymore, that comes from the lizard. And then making sure that at the right place, it's a GLP, a GIP, a glucagon, or these are shared amino acids anyway, so you don't have to worry about it. Not making too many changes remains important to prevent immunogenicity. So it turns out these triagonists are even better than dual agonists. Just look at the weight curve here. This is a diet-induced obese mice. This is a dual agonist, sort of like Mounjaro, tirzepatide. This is a triple agonist. It's pretty much twice as good. Same on food intake, on fat mass, what is really our target here. Cholesterol improves, blood glucose improves, glucose tolerance, insulin levels improve. and you also clear the liver of fat. This is relevant because as soon as you have a glucagon in the molecule, and it's in superior ways buffered with two incretins, GIP, GLP, you have receptors at hepatocytes that you're targeting. GIP and GLP don't target the liver directly. Glucagon does. That's why patients that may have a fatty liver disease within their metabolic syndrome will particularly benefit from versions of these dual triple agonists that have glucagon. Now, this also got published about 10 years ago, and now we have versions of this that are in phase 3 trials. It's actually 3 or 4 companies that are reaching or are in late stage phase 3. We assume that everything goes well, this could be approved in maybe 2 years or so. This is the version retatrutide by Eli Lilly again, and they have disclosed phase 2 data. You achieve 24% weight loss in the highest dose on average after only 48 weeks. And the trajectory is still pointing downward, so this might end up at 30% body weight loss. Now, in parallel, in the lab, we're still working on understanding mechanisms of triple agonists as well. So during the COVID lockdown, we made a mouse that is deficient for all 3 receptors, a triple receptor knockout for GIP, glucagon, and GLP. And when you give the triagonist to these triple receptor knockout mice, you see nothing, luckily, because you don't want to see anything. Otherwise, it would mean we sort of have a dirty drug that targets other stuff. It works in the wild type, it doesn't work in the triple knockout. So as I said, there's a bunch of these agents out there in clinical trials with many companies. It's probably more than the ones I put here down a couple of weeks ago at this time. I believe this is really important because we don't know enough to say which version will be the best. That will have to be figured out. And we need that competition in the future because these drugs are too expensive and not readily available for everybody who needs them. So having a lot of them more affordable, more available is going to be important. So the 30 years of history are summarized in this slide more or less. There was no treatment. If anything, was placebo effect. Then monoagonists got better and better before dual agonists really made an important step forward, and triagonists will get us to a place where these pharmacological treatments are sort of like a gastric bypass in terms of the weight loss benefits. But you can stop if there are serious side effects. You can adjust. You can, once they're better drugs, exchange. With a gastric bypass surgery, it's irreversible, in addition to being invasive. Now, this is take-home message of that whole part of my talk, saying dual and triple agonists are better than monoagonists, and this is getting us close to gastric bypass. Now let's think a little bit about what else this could do, and you may have reading if you are working in different parts of medicine, in different areas, about benefits of these drugs. They go beyond obesity. Do we have any there? Now, for example, cardiovascular benefits. That seems pretty obvious, right? If you massively improve metabolism, decrease food intake, decrease fat mass, you will have benefits for cardiovascular risk. It seems to go even beyond that. This will be really hard to dissect in the end. How much of that is a consequence of improved metabolism and body weight, and how much of that is direct effect? Some of these receptors of these gut hormones are actually in the cardiovascular system, and there are colleagues that believe there is significant contribution of direct effects. Now, other things, sleep apnea. Well, that is correlated with obesity, obviously, and if you decrease obesity, you decrease sleep apnea. Not clear if not central nervous system effects of these drugs do something there as well. Then kidney health. Cancer, colorectal cancer, other cancers are risk for those are decreased. We know obesity is a risk factor for types of cancers. If this is maybe a direct consequence, or GLP, GIP drugs are decreasing inflammation, and decreased inflammation is a good thing to prevent cancer, that all is not clear. Now, COVID is something that potentially you're less at risk at if you are under the treatment with these types of incretin-based drugs. Dementia, neurodegeneration, there seems to be interesting signal there. Parkinson even, maybe Alzheimer's disease. Still, the right studies haven't been done yet. We need way more information, but the size of the phenomena reported are sort of at the size where some of the best candidates for Alzheimer's disease currently are showing their effects. Addiction. Nicotine addiction is something that can be positively influenced. One reads in the literature. Now that makes sense because these food intake drugs, these body weight drugs that are based on incretin hormones, they're really working on the addictive component of food intake. Now, it's kind of obvious that that might work for other addictions like alcohol use disorder. Actually, the data for benefits fighting alcohol use disorder of these drugs are among the most corroborated and thorough data. I certainly believe that studies that are currently ongoing will show that this can be used against alcohol abuse. Now, we don't want to go as far and say this works against everything and we will live happily ever after, but that's certainly when you read the newspapers, you get the impression, oh, this is a wonder drug. It's not. It comes with side effects, can only be prescribed by doctors who know what they're doing, because there are certain diseases, if you have those, you can't have these drugs. So I'll end finally with a few speculations what that means for us and medicine and society. First of all, hopefully, if there's enough of these drugs, we can save lives. Many of us can prescribe these drugs. It's really doing fantastic things to these patients who have been waiting for many years for something that helps them in their fight against obesity. And then you will hopefully have an impact on type 2 diabetes, less type 2 diabetes occurring in the future in 10 and 15 years, which then will save additional lives. It will also change the need for insulin. How much insulin needs to be produced in 10, in 15 years if these drugs are available everywhere and you don't have obesity anymore, you will certainly have much, much less type 2 diabetes. Now there's an industry that is really worried about this, which is the fast food industry and all these companies making candy bars and potato chips and so on. They're already feeling this and they have crisis meetings where they are worried about what this will do to their economy. Now it turns out economy and capitalism are very flexible and dynamic. So Nestle, for example, has already figured that out and they came up with a new food called Vital Pursuit, and it's the ideal GLP-1 induced weight loss accompanying food, whatever that means. It's frozen, I'm sure it's not cheap, but it's the food you should take, says Nestle, when you're taking these drugs. Now, a couple other things. Airlines are calculating already that they will need less gas, so they will save money because people will weigh less. It seems a reasonable thought at some point. And then the UK, I think, just figured out that you can maybe give these weight loss drugs to unemployed folks, and that will make them more likely to come back to work. Steve, is that correct? This is something they... Have you been doing this? Right. But those were thoughts that apparently came up. Now, those of you following celebrities in Hollywood, they will have noticed that at the Oscars, some people who used to look like this now look like this. This may or may not have something to do with these drugs, but even that development of folks losing a lot of weight rapidly and it's showing in the face has created a new industry, which is a lot of surgeons that start a practice that does nothing but dealing with the Ozempic face, so that you sort of get these skin falls taken care of that occur when you lose a lot of weight quickly. Now, are we done yet with obesity drug discovery? I would say no. I would argue that major progress, important progress, but much more is needed. If you really look at extreme obesity, we may need for some patients 40, 50, 60% weight loss. We can't offer that. We have a problem with tolerability. A lot of patients actually stop the treatment because of side effects, nausea, emesis, because they feel, well, you know, this takes a big hit on my social life if I don't feel like a dinner and I don't feel like a beer or wine anymore. I feel better, I look better, but I really am not fun hanging out with my friends anymore. These are arguments that one hears. And then when you look at comorbidities, I find this really interesting. If you reduce body mass index from 40 down to 30 or 27 even, you're not necessarily removing all of the comorbidities. There's a significant number of comorbidities of obesity still there, and maybe those need to be targeted separately and efficiently. And this is something that we are working on now we have been working on for a while, putting maybe steroid hormones and small molecules on these peptides and targeting some of these comorbidities, inflammation, etc. That's not in the clinic yet, but certainly something we want to do in the future. So coming back to something that Jeff said, is this the beginning of the end? We don't think so. It's the end of the beginning of new drugs for metabolic disease and obesity. And we in biomedicine, in this field, as you have seen, frequently use knockout mice to test if something is causally relevant. So as a loss of function experiment, I'd say if we would remove Jeff from the field, which nobody wants to, but if Jeff was removed, that whole story that I just told you would not have happened. Thank you for your attention, and I'm happy to take questions.
Well, thank you very much, Matthias, for a stimulating talk. Questions?